maybe. So I apologize for the cold. I know it's not very comfortable. So today we are starting a brand new chapter. This is chapter 4. Obviously, we are not covering chapter 3, which is on embryology. I seldom, if ever, cover chapter 3 on embryology because it really should be an entire course in its own right. So if you have a chance, make sure that you read it just for your own information. This is not an embryology class. If you ever have a chance, take one. They're awesome. But whenever applicable, I will talk about some developmental things as they apply to things that we're looking at. Actually, we're going to mention a couple of those today. So we're jumping right into the next chapter, chapter 4, on tissues. I hope you read the chapter. It's super long. It goes on like for 200 pages, right? It's like, come on. It's like a nightmare. End already. But it's on tissues, and you might have probably guessed by looking at the slides, especially this first slide in Canvas. And here's where it says that we're going to, for chapter 4, concentrate on CPE connective tissues and epithelial tissue. That's not to say that we're not covering the other two very important tissue types. We're going to cover them, not only cover them, but in great detail. Muscle tissue, there's I think 2 chapters on that. And nervous tissue, that's the big one, 4 chapters on nervous tissue. You know, I'm a neuromuscular kind of person. I love muscle tissue, and of course, without nervous tissue, muscles wouldn't work. So we'll get to those. But in this chapter 4, we'll be covering just connective and epithelial tissues. The chapter covers all of them, I think, too quickly. But anyway, so that's the plan. When we talk about tissue, the first thing you want to think about is, what is a tissue? Well, a tissue is what? It's a bunch of similar cells, that perform a similar function. Last week we finished cells, the basic living units, right? And now we're going to put cells together to form these cohesive structures known as tissues. Remind me, please. Actually, we just reminded ourselves, but the 4 main types of tissues are epithelial, connective, muscular, and nervous tissue, right? We're going to start with epithelial tissues. They're super interesting, and epithelial tissues, by definition, do 2 things. They cover and they line, right? So we talked about how the skin, which is the next chapter, oh, bless you, next chapter on the integumentary system, that's your skin. But basically, the skin has 2 parts. The upper part, the epidermis, is a type of epithelial tissue. Yes, so it covers. And the other place they would find epithelial tissues would be as lining tissue. We talked about how the lining of the stomach is probably the most rapidly regenerating tissue. Those cells probably turn over every 3 to 7 days. That's really quick turnover. So cover and line. And epithelial tissues have some characteristics that you want to remember. First of all, they're highly cellular. What does that mean? A bunch of cells, very closely aligned. Lots of living cells, sometimes non-living, but highly cellular with very little intercellular material. Okay, so you could see on this diagrammatic slide, you could see all of these cells. What kind of cells? Epithelial cells lined up very closely aligned, right? Is there a space between them? Yes, there is, but not a big space. By the way, these spaces between cells are called either intercellular spaces between the cells or extracellular space outside of the cell. Either one would apply. Intercellular would be more specific between the cells. Now, these spaces, extracellular and intracellular, are filled up with a material that is non-living, right? The cells are living, stuff between them would be non-living. So again, high cellularity, lots of cells, not too much space or extracellular material. These cells also have a very high regeneration rate. What does that mean? They're constantly dividing, right? Remember we talked about how we have a brand new epidermis every 30 to 35 days, so they're constantly dividing. Lots and lots of cell division, mitotic figures. The other thing about epithelial tissues that's important to understand that they are avascular. What does that mean? Avascular, without blood vessels, right? Avascular, you're going to put an A in front, it would be without vascularity, without blood vessels. So they don't have a built-in blood supply. So oxygen and nutrients or whatever that do need to get to these cells get there by diffusion, right? Have to move along. So these are avascular tissues, but they do have nerve supply to them, and that is known as innervated with nerves. So for example, if you very lightly touch, your epidermis, even very, very lightly, right, you could feel it, right? Because this epidermis, epithelial tissue, has nerves. What is that called? Innervated, right? Now, don't ever do this, but you know what a water blister is? A little bumpy thing, right? It's got this fluid in there. So when you have a blister, it actually is a separation between your epidermis and the dermis. The dermis is always very vascular, right? But, and then never do this. But, you're probably going to pop that thing, right? So if you do, but don't, you get infected, you will not bleed because this tissue is avascular. Is it going to hurt? Oh, yes, ma'am, right? So think in terms of, these tissues being avascular but innervated. All right. The next thing to note is, since these tissues are avascular, they should be very, very close to a vascular source. And without exception, underlying or supporting all epithelial tissues would be a highly vascular tissue known as 3 letters CTP, which stands for connective tissues proper. And there's different kinds. So back to the skin example, right? We got the epidermis. It's avascular but innervated, right? And then deep to the epidermis is the one. Anybody? Dermis, right? And dermis is a type of connective tissue proper that has a specific name. All right. The other thing to note is a couple of things. So if these cells are very closely aligned, they have to kind of be closely aligned because they cover, protect, and so forth. The lateral borders of these cells have specialized functions, little zippers and Velcro things to keep them aligned together. So there's a lot of these features on the side of the cell, lateral side, lateral border.
Thank you.
And then the other thing would be that, remember we've got epithelium. Epithelium, deep to it would always be CTP, right? So what's between? Well, all epithelial tissues are sitting upon a non-living structure known as the basement membrane. Basement membrane, non-living. So the basement membrane, please note, has 2 parts to it. And the 2 parts of the basement membrane, I'll show you, I'll go over the pictures, would be basal lamina, again, non-living. Basal lamina would be a bunch of glycoproteins, proteoglycans. For the chemists in the group, doesn't mean you need to know for this class. But basal lamina would be these protein-like structures that are made, secreted, released by the epithelium. Okay, And deep to that would be a layer of reticular fibers formed by the cells of the connective tissue proper, right? So basement membrane is like a coordinated effort to form this structure. Basement membrane functions. Well, the basement membrane not only supports the epithelium, kind of forms like a scaffolding for the epithelium, and also to some degree it prevents pathogens from getting through, right? So it's like a little barrier, again, non-living tissue. Then we'll also note a couple of other things. We'll note that the cells, there's 3 of them, are said to be polarized, and by that we refer to that each cell border or side is slightly different from the other. So they're kind of specialized borders, or borders would be a good way of putting it. So first of all, the border that is kind of free, there's nothing on top of it. This border on kind of top is known as the free or apical border. Free or apical border. The borders are on the sides. On the sides where we talked about, that would be the what? Lateral borders, right? And this other term also makes sense, the basal border. The basal border is the bottom border of the cell. It's the one that's sitting on that basement membrane. Now, depending on what kind of epithelium it is, on the free or apical borders, you may or may not have specializations, specialized features. For example, if this epithelium is lining the respiratory system, right, which is a pseudostratified epithelium, right, and it has to have the ability to move stuff along on the apical border of this epithelium, you would have what, please? Cilia. Absolutely. Remember, cilia are composed of microtubules, and they beat or move in a very coordinated fashion to move things up through the respiratory system. Now, I know you probably know, smoker or two. If you be smoking, don't stop. But smoker's cough, you've heard of that, right? It's horrible and it's probably painful for the smoker. And that's because in smokers, those cilia are completely destroyed. So there's no way for the respiratory tract to be able to move all of the stuff along to the top, because all the stuff, the gunk within the large, things of the respiratory system, the bronchi, the tubes, the bronchioles, right? They accumulate mucus and so forth, and that needs to be moved up, not down, right? You don't want to get stuck within the alveoli. And it's that ciliary structures that help move that stuff along. Now this sounds gross. So all this stuff goes up to the oral cavity and you swallow it. Okay, when you have functional cilia, you don't really notice that. But in smokers, they don't have the benefit of that escalator. It's called the ciliary escalator, right? The escalator is broken, and therefore they need to forcefully cough stuff out. Okay. Now, if this epithelium does not need to move stuff around, but if it's in the business of absorption and secretion, then the apical border would have specializations known as what, please? Microvilli. Now, don't get confused with microtubules and microvilli. Micro just means small. Microvillus means small, little hair-like thing, right? And microvilli are typically shorter than cilia, microvilli, and they function to increase surface area. So there's more membrane over which things enter and leave. So where in the body might you find an epithelium that has microvilli? Small intestine, right? The small intestine, that's where you have most of the absorption. To be sure, other areas as well. And if you take a look at the apical border of one of the cells that line the small intestine, they're called enterocytes. Remember, I was in grad school. It was like a slow weekend. I was waiting for things to happen. They do that a lot in research. But I thought, oh, what am I gonna do? I've got several hours. So yes, what I did was I made a section of the apical surface of one cell that is lining the small intestine. Happens to be a rat, and I counted how many microvilli. How many do you think per cell? Over 3,000 per cell. And that cell only lives a few days. Isn't that crazy? Anyway, so microvilli increase surface area. They are not composed of microtubules. Think of microtubules in movement. These things don't move. They provide more membrane over which things enter and leave, absorption and secretion. And then finally, the lateral border on the side, the lateral borders of the cells have specialized structures that hold these cells together. Please note the following. Going sort of from superior to inferior, we first have tight junctional complexes. And the book gives you a lot of detail on that, but essentially, these tight junctional complexes serve to hold the cells together by virtue of the fact that the neighboring cells, these would be the tight junctional complexes, which have proteins. Remember, cell membranes got phospholipid bilayer, right? We got proteins going on. And in tight junctions, the proteins from neighboring cells are fused. And these fusion sites, or tight junctional complexes, is more complicated than this, right? But they are going all the way around the perimeter of the cell. Okay, so that's like a zipper going all the way. I think the book uses zipper for something else. Let's call it a Velcro band all over the cell, all around the cell, and that connects one cell to the other. Tight junctional complexes form somewhat of a permeability barrier, keep things from going through very, very quickly. That's not to say that epithelial tissues are impermeable. Some stuff does get in, but kind of mitigates too much stuff getting through. Then we have, usually inferior to the tight junctions, these cool structures right here, and these are called desmosomes. Desmosomes. Desmos means binding. Soma, body. Binding bodies, right? So if the tight junctions go all the way around the cell, right, the Velcro thing, it keeps them together, then the desmosomes are little buttons, little spot adhesions that strengthen cell-to-cell adhesion. It keeps the cells kind of from tearing apart from each other. And they have these little linker proteins, or these are little buttons. So desmosomes are also there to maintain the integrity of the cells, keep them from separating. And then finally, these really cool things usually appearing towards the inferior portion of the lateral border of a cell. These are called gap junctions. And gap junctions, here we go, they're composed of proteins from adjoining cells, and these are called connexon proteins. They connect. Think of connecting connexons. And in the center of these connexons, there's a channel. So you could see how gap junctions, look at that, they connect the cells via these little channels through the connexons. So that allows cell-to-cell communication, right? Cell-to-cell communication. So what are they doing? Are they texting each other? Kind of, right? Because it goes real quick. But what they're texting would be ions, charged molecules, so to coordinate an effort of the entire epithelium. Speaking of a coordinated effort of any of these structures, we're talking about the tight junctional complexes, the spot desmosomes, and the gap junctions as being specific to epithelial tissues, which they are, but they're also present in other cell types as well. For example, in cardiac muscle cells, when we have a whole chapter on that, there are many of these gap junctions between myocardial cells. How would that help that cell? myocardial, cardiac muscle cell, to very quickly get that information via charged molecules. So the cells are communicating, texting very quickly to ensure a unified contraction. I know some of you are nodding because you're taking physiology. You'll cover all of this. So those are the lateral borders, and we talked about the basement membrane as well. So to summarize, the epithelial tissues as a whole, right, they're highly cellular. Or do they have a blood supply? Nope. What do you call that? Avascular. Do they have a nerve supply? Yep.
Yes.
That means they are innervated. Do they undergo lots of cell divisions? Yep.
Yeah.
So high regeneration rate. Are they polarized? Yep.
Yeah.
So what do you call the top of the unattached layer? Free or apical surface? We could have microvilli, we could have microtubules, or we could have nothing. It depends on where that epithelium is. Then we have the lateral borders, right? And the basal borders of these cells are resting upon a non-living, what please? Basement membrane, basal lamina plus reticular fibers. It's a joint effort by these two tissues. And always without exception, deep to or on the other side of the basement membrane, you always have connective tissue proper, which is highly, highly vascular, right? And that's where epithelial cells get their oxygen, nutrients, and things like that. This is just a cool picture of semi-longitudinal, semi-cross-section of one cilium. So cilium one, cilium more than one, singular and plural. And at the core of each cilium, you have a very specific arrangement of these miniature straw-like rods called what, please? Microtubules. Now, where else can you see microtubules? We talked about this on Wednesday. Centrioles, right? Centrioles are also composed of microtubules in a different arrangement. The arrangement of microtubules in cilia is said to be 9+2 arrangement, which means we have 9 little doublets on the outside and 2 single ones in the middle. Right, slightly different arrangement. And during movement, I mean, we have people studying the dynamics, the physics of this, but there is an interaction between the microtubules that causes them to slide past each other, right? Cause them to slide past each other. All right. Then, what do you call the apical surface specializations that do not have microtubules but increase surface area, please? Microvilli. See, I caught myself on that. So microvilli do not have microtubules. So no microtubules are not going to be moving, right? But they simply increase surface area. There's a very specific arrangement of them at the bottom. If you read more, there's a basal body, kind of coordinates stuff. We don't need to know that. All right, so we are anatomists, and anatomists like to classify stuff. Medical personnel likes to classify stuff. So when you think in terms of what could go wrong with epithelial tissues, for example, skin cancer is still the most common type of cancer as far as I've read. And if we talk about basal cell carcinoma or squamous cell carcinoma or malignant melanoma, you would then, as a medical professional, know what kind of epithelium is involved, right? So we need to classify stuff. When you talk about classification of epithelial tissues, there's 2 things that you need to note. First is how many layers or how many rows of these cells are there. There's only one layer or one row, it's referred to as simple. If it's more than 1, 2 or up to in the epidermis, it could be like 50 layers, right? Then that's called stratified. 1 row simple, more than 1 stratified. Strata means layers, like in the Earth's crust, right? Then the other thing to note is the shape of the cell. So they come in 3 different varieties. First of all, you have a flattened cell that looks like a fish scale, and that's known as a squamous or squamous cell. I think squashed, right? Very, very flat. And typically, the nuclei of these cells kind of follow the shape of the cell. So in squamous, squashed, flattened cells, the nucleus would be kind of flattened, slightly elongated, right? Then if you have a cell that is more or less square-shaped, of course these are 3-dimensional things, right? We call these cuboidal cells, cuboidal. And here's the nucleus. Shape it could vary from one type of epithelium to the other, but the nuclei are kind of roundish, maybe sometimes ovalish. They could be in the center of the cell or towards the bottom, typically in the center, not always. Then if we have these tall cells are taller than they're wider, they look like columns. Those would be what? Columnar cells. And typically the nucleus is kind of ovalish, and it's usually, not always, but typically towards the bottom of the cell. So we've got squamous, we've got cuboidal, and we've got columnar. Now, let me then look at this. So, one layer simple, these are the cells, they're sitting on a basement membrane, CTT on the other side. So, what would you call this epithelium? Simple, squamous. Now here's another one. Here's this one. So definitely more than one layer. So it would not be simple, but stratified. But then we have a situation here. So we've got like more or less a squamous cell on top, right? And these are more cuboidal. So is this stratified squamous or stratified cuboidal? Squamous, exactly right. So to make sure that everybody's on the same page, in identifying, defining epithelial tissues, classifying them, we always look for the shape of the cell at the apical border, at the top. Doesn't matter, typically in a stratified epithelium, there's different types of cells. One more time, in defining or classifying epithelial tissues, first the number of layers, simple or stratified. In stratified epithelia, when you're identifying the cell, you look for the cell at the top, at the apical border, not on the basement membrane, right? So this would be a stratified squamous epithelium, top layer, furthest away from the basement membrane. All right, so now we're going to identify different types of epithelial tissues and try to figure out where in the body they are located. First of all, what would you call the thinnest type of epithelium? Thinnest, simple, squamous. One row of flattened cells. That's the thinnest epithelium. One row of flattened cells. So this epithelium would be really good at diffusion. It doesn't have too many layers for stuff to get through. Things enter and leave fairly nicely. Locations. The book gives you tons of locations, but I try to streamline. Somebody emailed me over the weekend. It's like, oh, there's so much stuff. Are we going to cover everything? Sadly, no, unless we're here for another 5 years. Nobody wants to be here for another 5 years. So I kind of pick and choose what is the most common, the most representative. Details, you'll always be able to Google that stuff. Don't believe everything on Google, by the way. So anyway, locations, just the ones, and this is plenty, right? Just the locations that I've indicated, they're also discussed in the book. And where would you find a very thin type of epithelium? The alveoli. The alveoli would be the little air sacs within the respiratory system, right? So when we breathe, we're obviously, we need to bring oxygen to all the body cells. We're breathing out carbon dioxide, that sort of waste gas, right? So where is that gas exchange occurring? Obviously in the lungs, specifically between the alveoli, the air sacs of the lungs, the very end points, right, and the surrounding blood vessels. That's where oxygen, carbon dioxide, are exchanged, gas exchange. So that makes sense, right? Then we also have a specific type of simple squamous epithelium that would be lining blood vessels and lymphatic vessels and the heart. And in that case, it's called the endothelium. So simple squamous is found in a bunch of different spots. Flashcards, please. That would make your life, I think, a lot easier. So anyway, what are the smallest blood vessels in the body called? Blood vessels. Capillaries, right? So we've got this cardiovascular system. We've got the heart. Blood vessels that carry blood away from the heart are called what? Arteries, right? Then they get to be smaller arterioles. Then they become the smallest capillaries, right? And then back to the heart, venules, and veins. You've got that circular path, right? So what happens at the level of capillaries? That's where gas exchange occurs, right? Between the capillaries and the body cells. Therefore, there's that simple squamous epithelium, specifically known as the endothelium. Now, a similar simple squamous endothelium also lines the other blood vessels. It lines the lymphatic vessels, and it lines the heart as the endocardium. There's more stuff in there, okay? Then, remember that we have serous membranes. I got another email that I think was a really good question regarding serous membranes. Serous membranes, let's see. There are 3 areas defined by serous membranes, all that have names that begin with a P for the heart, pericardium, serous membranes for the lungs, plural, for anterior abdominal, peritoneal. Serous membranes, how many layers? 2. The outer parietal, and the inner one would be visceral. Little space between the two contains the serous fluid, right? Serous fluid. And each one of those membranes, let's say parietal and visceral, are composed of that simple squamous epithelium. In this case, it's also called starts with an M, mesothelium, and a little bit of CTP. Don't forget there's always CTP in there. All right, that's always the arrangement. So mesothelium is that epithelium. What kind? Simple squamous epithelium that lines these ventral body cavities. Pericardial, pleural, peritoneal, serous membranes, right? Mesothelium. Now, middle epithelium, but doesn't matter the etymology, but have you heard of mesothelioma? Mesothelioma, they're still doing little ads on TV. Lawyers want to get your cases. Mesothelioma, which is a really scary kind of cancer, typically it's caused by asbestos. And those little particles get stuck, and the asbestos then changes, alters some of these serous membranes into a cancerous condition that's known as mesothelioma. And typically it goes to the lungs. Could it go to the heart? Yes. Could it go to the peritoneum as well? So anyway, try to get some clinical correlations if you can. And whenever I'm familiar with them, I'll be happy to share those with you. The last part of locations would be, now this is still simple, squamous epithelium would be within the kidney. That's where things occurred initially, filtration. I know some of you have already had physiology. And these structures are known as glomeruli. Kidney glomeruli, also lined by that simple squamous epithelium. Okay, that's just called simple squamous epithelium. So it's got a lot of applications, a lot of places you could find simple squamous epithelium. So it's going to kind of get easier because look at that location. I'll just give you one. As far as the next type of epithelium, one row square cells, name the epithelium, please. Simple cuboidal epithelium, right? Do you find it in different places? Absolutely. But the most common spot would be kidney tubules, not the glomeruli. That would be simple squamous, but kidney tubules. Now let me explain this slide to you because we need to figure out what we're looking at. Kidney tubules. So here's kind of a semi-longitudinal lengthwise picture, right? And these are all cross sections. So think of tubes, right? So they're hollow tubes. Inside you have renal filtrate stuff, right, before it becomes urine. But if you look at these kidney tubules, first of all, please note this word lumen. Lumen just means the opening, right? So you could have a lumen within the center of blood vessels. That's a lumen, right? In that case, it contains blood. In this case, the lumina of kidney tubules would contain renal filtrate. That's for the future, right? But hollow tubes, right? They contain stuff. The opening is the lumen. But then what we're looking at would be the cells. Now, this is not a diagrammatic, so this is what it really looks like. So, a couple of things. You might not be able to see the boundaries between the cells. They're very, very thin. Typically, depending on which stain you use, typically, the nuclei do pick up more stain, more color. Remember, this would be a light micrograph. And so you can see the shape of the nuclei. So they're kind of roundish, so probably, simple cuboidal epithelium. And you might not be able to see the boundaries. Simple cuboidal epithelium. And the lumen would be the opening. Now, if you have one row of tall cells, that is called a simple columnar epithelium. And here, simple columnar epithelium, let's see. Where might you find it? Well, most of the digestive tract is lined by a simple columnar epithelium. The uterine tubes, simple columnar epithelium. The lining of the uterus, also a simple columnar epithelium. Now, depending on where we're talking about the location, right? If you're looking at that simple columnar lining of the gastrointestinal tract, of the small intestine, what apical surface specializations would really help in that region? Would be absorption and secretion. So at the apical surface, you would have microvilli, right? Because we're absorbing stuff. To be sure, secreting some enzymes as well. So look for those specializations. So midterm question might be what kind of epithelium specifically lines most of the digestive tract? It would be a simple columnar with microvilli, right? But if I ask you specifically what kind of epithelium lines most of the respiratory system, we talked about smokers and some of those things kind of falling off, right? It would be a ciliated simple columnar epithelium. So again, each part of the body is specialized to do what it has to do. And to enable those epithelia to do their jobs, it would either be microvilli, absorption, secretion, or a ciliated type of epithelium. Okay? And the uterine tubes, also known as fallopian tubes or oviducts. We also have cilia over here. And how would that help in the fallopian tubes, uterine tubes, oviducts? What's happening in there? We got the ova moving along, right? So you need to make sure that although if fertilization is going to occur, It typically occurs within the oviduct uterine tubes. Implantation, however, occurs within the uterus. Otherwise, you have an ectopic pregnancy. I have a whole chapter on that. Okay, so I misspoke about the cilia. The lining of respiratory system is ciliated, but it's not simple cuboidal. I'll come back to that. Now, we could also talk about striated epithelia, more than one layer, right? More than one layer. And in the case of striated epithelia, what are your choices? We have stratified squamous, we could have stratified cuboidal, we could have stratified columnar. Please listen carefully. Stratified columnar and stratified cuboidal are very, very rare. And you read the book, it indicates that. Don't worry about them. Stratified squamous is actually fairly common. Stratified squamous, many, many rows of cells. The cells at the top border are what shape, please? Squashed squamous, right? Now, stratified squamous epithelia could either be super hardy and kind of waterproofy because the cells have a lot of waterproofing kind of proteins known as keratin, right? So when you need more protection, you have a keratinized stratified squamous epithelium, and that would be the epidermis. Keratinized stratified squamous epithelium. Keratin would be a waterproofing thing, right? So you could go into any Walgreens or CVS, whatever, and you look at products, which I enjoy doing, but it kind of also cracks me up because it says, oh, this is it has keratin in it. So it's not going to absorb probably, but might smell pretty. And if you want to buy it, a lot of money, do it. It's probably not going to help that much. Anyway, I know I burst your bubble over there, right? You have all the products. You don't need that, all the fancy stuff. Anyway, so the epidermis is what kind of epithelium? It starts with a K. Keratinized stratified squamous epithelium. However, lining of the mouth, lining of really most of the cavities that open to the outside immediately would be a less bumpy, less well-protected, non-keratinized, stratified squamous epithelium. So obviously, I mean, you don't want to touch your face, but on the outside, it's kind of rougher because of the keratin. Lining of the mouth, much, much smoother. No keratin, non-keratinized, stratified squamous epithelium. Okay. Then we've got this stuff going on. Let me see what we have here. All right. So one more time, the stratified epithelia, stratified cuboidal, columnar, rare. Don't worry about them. Stratified squamous, yes, keratinized and non-keratinized. Now, what's happening here? Well, another type of epithelium. It's really pretty much stratified-ish, but it's called transitional epithelium. Transitional epithelium transitions in shape to accommodate for volume, right? And this type of transitional epithelium, again, when the cells are kind of pulled or stretched because that structure is filling up with stuff, it becomes a thinner epithelium. When that area doesn't have the pressure on it, it becomes more bumpy. It accommodates for volume. Therefore, where do we need to accommodate for volume? Well, a lot of the urinary system regions, specifically, and don't get the words mixed up because there's a uterus and there's ureters or ureters, right? So when you look at the two kidneys, here they are, and here's the urinary bladder, right? These tubes are called the ureters or ureters, right? And they are lined by that transitional epithelium, accommodates for volume. Just so much, right? And then urinary bladder is also lined by this transitional epithelium. When your bladder fills up with a lot of urine, it causes pressure. You need to go void that out, but accommodates for a little bit, gives you a little bit more volume. And then, what do you call the tube through which urine ultimately leaves the body? And that's called the urethra, also lined by this transitional epithelium. Okay, then we can take a look at this. So I misspoke about the ciliated. The lining of the respiratory tract is ciliated. We talked about cilia and smoking and stuff like that. By the way, if you're a smoker, live with a smoker, no bueno. But if that no longer is the case, will those cilia grow back? You stop smoking? Absolutely. Because this is highly mitotic cells that keep reproducing, right? It might take a while, right? Because it depends on the damage and all that. So pseudo, the prefix pseudo, the P is silent. Pseudo means falsely. So falsely stratified. So kind of looks stratified, but it really isn't. So pseudostratified epithelia, definition, contains short and tall cells. So the nuclei kind of look like they're layered, but they're not. Because all cells of the pseudostratum epithelium, you've got the short ones, you've got the tall ones, they're all sitting on the basement membrane. But in some of these preparations, using microscopes and stuff, it gives you the illusion of many layers, but it's only one. Pseudostratified epithelium. So midterm question might be pseudostratified epithelia. How many rows of cells? One, right? But we have short and tall cells. Typically the short cells undergoing cell division, then they grow, right? And then they get sloughed off or what have you. And in the case of the lining of the respiratory system, what do we have on top?
Cilia.
We've got cilia, because they're moving stuff along. Couple of other things. We haven't talked about glands. What is this thing right here? Here's the one right here. Maybe this one. These are goblet cells. So goblet cells, okay, they kind of look like this, like goblets, like wine glasses. So, you've got the liquid here, and here's the stem of goblet cells. And goblet cells are examples of a single-celled exocrine gland. Goblet cells produce this protein compound known as mucin, and then the mucin is secreted out. What do you call this process of release using vesicles? Exocytosis, right? So these vesicles exocytose mucin. And then what's outside of all these cells? We're 64% water, right? So once that mucin mixes with water, it forms that viscous, sticky stuff that we know as mucus, right? So goblet cells produce mucus, essentially. So if you take a look at this slide right here, pseudostratified, yep. Ciliated, and with goblet cells. And here's that trachea and other parts of the respiratory system. Okay. By the way, in lab, we will be looking at some micrographs of epithelial tissues. I will never, ever ask you to identify a pseudostratified epithelium. There's no way you could, after many years, but the best way to figure that out is to do serial sections to follow each cell down to the basement membrane. So again, pseudostratified epithelia, kind of know them in passing, or if I show you a picture of a pseudostratified epithelium in lab, I will tell you it's pseudostratified. Then how many rows of cells? One. Short and tall cells. What's on top would typically be the cilia. Perfect. Okay. All right. So speaking of glands and stuff like that, glands. Glands start out, developmentally speaking, as epithelial tissues. And then they become more specialized because they start to secrete stuff, make and secrete things. So glands are structures that make and secrete stuff. There's two different types of glands, and those would be endocrine and exocrine glands. Let's talk about endocrine glands first. Endocrine inside these glands, endocrine glands do not have ducts. They secrete their product directly into bloodstream. Please listen carefully. Endocrine glands make and release only one kind of product. Hormones, exactly right. To be sure, there's tons of different hormones, right? There's estrogen, progesterone, and there's insulin, and there's glucagon, and what have you, right? But hormones are blood-borne messengers, right? So endocrine glands, ductless, they secrete into the surrounding bloodstream, and the only product that they make would be hormones of different types. Okay? So those would be the endocrine. We have a chapter on that. I may or may not get to it because that's really physiology. But exocrine glands. Exocrine glands would be glands that secrete all sorts of stuff except for hormones. And depending on whether there's many cells or one type of a cell, whether they are branched or not, you could classify them. I'm going to make things easier for you. So exocrine glands, they secrete their product through a system of ducts. The ducts might be branched or unbranched. Doesn't matter for us right now, okay? But I do want you to know the morphology of the secretory units, the parts that actually make that product. And this is pretty easy. So the secretory products all either look like test tubes tubular, they look like tubes, or they look like little circular type things, and this is known as alveolar. Alveolar means grape. That's one of the several terms that I really don't like. Why is that? Because there are alveoli everywhere, right? We talked about the lung alveoli, right? Already used that term. They kind of look like grape clusters. And then here we have this kind of alveolar grape-like structure. So exocrine glands, do they have ducts? Yes. They could be branched or unbranched. We don't care. But the morphology of the secretory units, either like a test tube, tubular, or like grapes, that would be alveolar. And most of these are multicellular, many cells. And then finally, back to this one. The only example of a unicellular, one cell, the only example of a unicellular exocrine gland would be the wine goblet cells, right? Only example of single-celled. Do you have a question or? No, you're talking, okay. Yeah, if you can kind of keep it down because remember I'm talking through a mask. So I'm trying, can you guys up there kind of hear me? Okay, so I'm trying my best, but please don't talk because then I have to, you know what, I'm trying to resuscitate me. Anyway, goblet cells make what? Mucin, which becomes mucus. All right, now, we are done with epithelial tissue. As always, write down your questions. We'll address everything either at the end of a lecture, better even would be in lab. So let's get through this next part. And that would be connective tissues proper. Again, chapter 4, what are we talking about? Epithelium, been there, done that, and CTP, connective tissues proper. But we need to step back and talk about connective tissues as a whole, not just CTP. So what other types of connective tissues do we have? You've read the book. We've got bone tissue, cartilage. What's the only liquidy thing that we are talking about? Blood. So we got bone tissue, connective tissue, cartilage tissue, connective tissue, and blood is also a connective tissue. It connects things. But we also have this Other type of tissue that has a bunch of subcategories, and that's called connective tissue proper, which is a weird name. Is it proper? Is there an improper? I don't know. They're called CTP. You might have never heard of CTP. Have you heard of a tendon? Yep, ligament. Anybody watch the game yesterday? Oh my gosh, no, don't want to talk about it. But anyway, the Brock Star, what did he do? He pulled something in the elbow, was it? Yeah. Was it they talked about some tendon. Okay, so you've heard of tendons, you've heard of ligaments, right? Those are types of connective tissues proper, right? Another thing might be familiar would be adipose tissue. It's a type of connective tissue proper. So again, we got connective tissues, different types, blood, bone, cartilage, separate chapters on that. We're going to go into that. Bone tissue is absolutely stunning. Cartilage tissue, don't get me started. Blood is amazing, right? But here we're talking about CTP, things like tendons, ligaments, adipose tissue, and so forth. Now, first, let's take a look at and compare epithelial tissues and CTP. So CTP from this point on. Which type of tissue, epithelial or CTP, has more cells per unit? More cells, tightly packed. Epithelium, right? Very little stuff between the cells. Look at this diagrammatic of a loose CTP. Do we have cells? Yep. And they're not very similar, right? They're kind of spread apart. What's between these cells? Those are little bit things. Would be between the cells. So we've got the cells. Look at that. We've got all the cells. I put them in red for you, right? Got all the cells. Those are living things. The stuff between the cells is known as a matrix, like the movie, okay? Which doesn't mean anything that much. But anyway, so matrix. Matrix would be the extracellular stuff. Is the matrix living tissue? No. It's stuff that is secreted by the cells, but it's non-living. It's non-cellular, okay? So it's sort of like you have these cells. If you think the cells as little vegetables and vegetable soup, what have you. But the brothy area would be the matrix, non-living. Now, the matrix has two subparts to it. It's got ground substance, which would be like a semi-fluid gel. And that would include like little ions, and it would include lots of water, right? Some molecules that maintain the water inside there. So that's kind of the brothy stuff. And within this ground substance are embedded miniature little rods. And these are called fibers. Fibers. Ground substance is the semi-fluid jelly stuff. And then within it would be these also non-living little fibers. Now, you've heard of collagen, right? So many parts of our body are collagen. So what kind of fibers? Well, we could either have collagen fibers. Those are the big ones. They look like big old ropes, right? And each collagen fiber, bunch of ropes, like a big old rope, it's made up of individual little strings, right? And each individual string is known as a collagen fibril. Big old fiber, collagen fibers, right? Big old ropes. Those maintain the integrity of the cell, allow for some straightening of the tissue, but the collagen fibers cannot, they could straighten, but they could not stretch. So it's like a rope that not a rubber band. We're talking about rubber band. So we've got a rope that's kind of curvy. So you could straighten it. Can you stretch it? No. Eventually it will rip, right? And yes, let's go into scars and stretch marks, right? So one of the lovely side effects of pregnancy would be stretch marks. Why? How does that occur? Well, when the skin stretches, right? Skin stretches, and there's collagen fibers in the skin as well. So straight. Okay, my son was 10 pounds. I remind him every day. Anyway, so I know, right? Yeah. Anyway, he's wonderful. So okay. So everything stretches, right? So those collagen fibers straighten, but then they rupture. Oh, I know. Adopt, right? And so then what happens? Then scar tissue. So these collagen fibers will start to repair themselves, and they form the scars, right? Battle scars. All right. So those are collagen fibers. Then we've got the reticular fibers. These are skinny little, not big fibers, but individual little fibrils. They're also collagen, a type of protein known as collagen. Now, I know a lot of you are taking chemistry. You're taking biochem or something like that, or OChem, there's different types of collagen. Okay, that's a whole other thing. Anyway, so then we also have, do we have rubber bands in there? Yep. And these are called elastic fibers. And elastic fibers are made up of a different protein known as elastin, so that's easy to remember. And these fibers do stretch and recoil. They stretch and recoil, right? And all these are present in the skin as well. So I had to take a picture, my selfie for my new ID card, right? And I'm thinking, oh God, this ain't cute. But as we get older, oh yeah, don't get me started. So anyway, the amount of elastic fibers in the skin, in the dermis, not the epidermis, in the dermis, slows down. There's less rubber bands. So I remember wake up in the morning and I scare myself. Like, what is that? Little pillow marks? You know what I'm talking about, right? Well, my granddaughter, who's 4 years old, she got what happened to? She's got more elastic fibers in there, right? So things happen as we get older. Oh yeah, we'll talk about skin. That's gonna be fun. Anyway, so what kind of cells do we have here? Now, in all types of connective tissue, right? We typically have the cells, and the outer stuff, the non-living stuff, is called, one more time, please, matrix, which contains a semi-fluid, ground substance. Embedded in that would be up to 3 different types of fibers. This is all the non-living stuff, right? So the question is, well, who makes most of the matrix? So it's there. How did it get there? And the answer is each type of connective tissue typically has kind of a builder cell. In the case of most CTPs, the builder cells are the what, please? They build fiber builders. Can you find something like that? How about, oh, look at that. Oh, it's right here. You got it. See, look at that. Fibroblast. Now, when you see a cell that has something blast, those cells, B for builder. If you remember that, that will help you. So fibroblasts build not only the fibers, but allow for most of the ground substance to be built as well. In future chapters, a bone matrix builder cell would be an osteoblast. Cartilage builder cells would be chondroblasts. But we're not talking about those yet. So we have fibroblasts. So midterm question would be, which cells build most of the matrix? Fibroblasts in CTP, right? We have macrophages. What do you think macrophages do? Phagocytosis. These are the big eaters, macrophages. Exactly. They undergo phagocytosis, and they're very large. They move around. They scavenge around, pick up things, and destroy things like pathogens or other dead or dying cells. Then we have a couple of blood cells. We have lymphocytes. Blood cell neutrophils are also white blood cells. We have the cells that everybody loves to hate, but we need them. These are the adipocytes. Adipocytes, adipose tissue. Fat tissue, very important for our survival. And then we have this other type of a cell. This is known a mast cell. Mast cells are involved in the inflammatory response. So they secrete histamine and heparin. The type of cells varies from one type of CTP to the other, but these are some of the basic ones that you will find. Okay, so mast cells secrete histamine and heparin. I'm allergic to pollen, I'm allergic to a bunch of stuff. So when my allergies act up, I take a what? Antihistamine, right? And that will slow down the progression of the inflammatory response, which is important to an extent, right? Heparin, how many of you work in a clinic or hospital, deal with stuff like that, deal with IVs? No? Heparin, does that sound familiar? Heparin, a heparin lock, Heplock, right? Have you heard of that? So if you are going to, have an IV in your patient, right? But then maybe they don't need the IV for a while, but they might need it again. Rather than keep sticking them, you leave one of the lines in and you put a what on? Heplock, heparin lock, right? And that prevents the blood from coagulating. So if you need to go back again and hook them up to something, it's already in there. You just add whatever else needs to be added. So heparin is an anticoagulant. Exactly how it works here, I'm not quite sure. All right, so now all we have to do is talk about the different types of CTP. Because remember, we are anatomists and we like to classify stuff. And I'll make it easy for you. So, a little bit of embryology. The embryonic type of CTP, before it becomes dense regular or adipose or dense irregular or elastic, is known as mesenchyme. All you need to remember is mesenchyme embryonic CTP. This is kind of loosely arranged, but we don't care. Mesenchyme embryonic CTP. It's kind of your stem cell type analogy, right? So mesenchyme could become any other type of CTP, embryonic, right? Kind of pluripotential. Mesenchyme embryonic, that's all we need to know. Now, the next type of epithelium, CTP, is called loose areolar. So we've got loose and we've got dense CTP. And the classification is kind of difficult, so I'm going to make it super easy for you. Loose areolar CTP, loose areolar. The loose has to do with the fibers being loosely arranged. So they're not coming together real tight. They're kind of spread out. Remind me, what are the three types of fibers? Collagen, reticular, and I wish I had more what? Elastic fibers, right? Those are the fibers. When these fibers are kind of loosely arranged, you have loose areolar CTP, right? Areolar means a lot of like spaces between, lots of matrix between the cells. And loose areolar CTP is the most common type of CTP. Loose areolar, we're going to clump it together. Loose areolar CTP is the most common connective tissue proper in our bodies. So where would you find, if it's so, prevalent, where would you find loose areolar CTP? It's your packing material. I don't think Amazon uses those little bubbly things. What do you call those little popcorn things? Isn't that really bad for the environment? So what else? But they have no more packing material, right? But they used to. They have a big one. Okay, so back to the popcorn days. So packing material, that would be your loose areolar CTP, in and around organ capsules, supporting a lot of membranes as well. Loose areolar CTP, all three types of fibers, loosely arranged, present pretty much everywhere. Functions, cushions, holds tissue fluid. Where's the tissue fluid found, please, folks? Within the ground substance, right? And also it actually has a lot of macrophages because check this out, kills pathogens, bacteria, viral particles, and such. Then here's this tissue, the one that we all love to hate. We all want to get rid of fat. Folks, without fat, we wouldn't survive, so I'm embracing mine. Adipose tissue is a type of CTP. Yes, it's kind of loosely arranged, but we're going to keep it simple. Adipose tissue, fat tissue, right? The main cell here, by the way, the main cells are the adipocytes, right? And they're full of fat droplets or one big droplet. So therefore, the nucleus and most of the cytoplasmic stuff is kind of pushed to the side. Here's a little nucleus pushed out. What happens when you lose weight? Do you lose the number of adipocytes? Do you have less red cells? No, each cell gets smaller. Unless you have liposuction. Don't do it. It's not cute. That's a different thing. I had a friend with liposuction. She's still hurting. It's been like 6 months. Don't do it. Don't do it. Exercise, whatever. Anyway, so, but adipocytes are really important. Now, where are they found? Well, look at that. They have a lot of fuel reserve. They're protecting all of that. Where do we carry most of our fat? Deep to the skin. Exactly. And like the stomach, like Travis was saying, the stomach region, that's visceral fat, right? So let's get back to the skin. We've got the epidermis. Epidermis, what kind of epithelium starts with a K? Keratinized, stratified, squamous epithelium. Deep to that is the highly vascular dermis. Deep to that is the what? Hypodermis, also known as is it up there? Subcutaneous tissue. So hypo, under the dermis, that's the Greek, right? Component. Subcutaneous would be the Latin. But check this out, it's also known as superficial fascia. A lot of you are going into kinesiology and stuff like that, right? So you talk about fascia. Typically when you talk about fascia, you're not talking about superficial fascia, you're talking about deep fascia associated with muscles and so forth. But superficial fascia, fat tissue, what's another word for this fat tissue? Hypodermis or subcutaneous tissue. This is all adipose tissue. One more minute, get your money's worth. Okay, so where else would you find adipose tissue in the body? Is it on here? Mm-hmm. The breast tissues, right? Kidneys and orbital fat. So our eyeballs are very nicely protected within their sockets with fat. Kidneys are surrounded by fat tissue. If you have a patient who loses a lot of weight, unhealthy fashion, and all of that fat diminishes, what's the first thing you'll notice about their eyes? Kind of deep set, right? And the kidneys, worst case scenario, the kidneys, the renal fat would also disappear and the kidneys start to kind of lower down. Okay, so enjoy the fat. So on Wednesday, we're going to finish up this chapter, and we're going to go right into chapter 5 on the skin, which ties right into this as well. All right, good job.